After that, we have discussed about other different types of organelles in part 1. Today, we continue with the study of peroxisomes. Peroxisomes are similar to lysosomes because they are membrane-bounded vesicles that enclose enzymes. However, the enzymes in peroxisomes are synthesized by free ribosomes and transported into a peroxisome from the cytoplasm. It means that, as you remember, we have two types of ribosomes depending on their location. Some are free in the cytoplasm, others are fixed on the rough endoplasmic reticulum. Now, free ribosomes are going to synthesize enzymes that will be packaged in peroxisomes, while bound ribosomes, and when we say bound ribosomes, we mean the ones which are fixed on the surface of rough endoplasmic reticulum. These ones are going to produce enzymes that will be later packaged into lysosomes. Now, what is the mission of enzymes packaged in peroxisomes? These ones are going to be used in the process of detoxification of hydrogen peroxide. Hydrogen peroxide is a poison, it is harmful, and it is produced during some metabolic reactions. So in order to protect our cells against this poison, an enzyme packaged in peroxisomes will be used in order to break down that poison into harmless substances, which are water and oxygen. Peroxisomes have varied functions. The first one has been discussed about. They contain the enzyme catalase, which breaks down hydrogen peroxide into water and oxygen. This has been discussed above. Second, they are especially prevalent in cells that are synthesizing and breaking down lipids. Third, in the liver, some peroxisomes produce bile salts from cholesterol, and others break down fats. Remember that bile salts are very, very important in the digestion of lipids that takes place in the small intestine, and these bile salts are temporarily stored in what we commonly know as the gallbladder. Vacuoles. Let us begin with the plant vacuoles. Plant vacuoles are the most conspicuous organelles. One large central vacuole in plant cells may take up to 90% of the volume of the cell. Conspicuous means very visible. Very visible. That's say vacuoles are the most visible organelles, but here remember we deal with plant vacuoles. When it's time to talk about animal vacuoles, the situation will be different. The central vacuole is filled with a watery fluid called cell sap that gives added support to the cell. Plant cells can obtain their support because of that watery fluid found in vacuoles. For example, you know that some plants present a weak stem and flexible. A weak stem and flexible to mean that they cannot adopt an upright position. Now, when a plant absorbs too much water from the soil, that time it can now have an upright position and maximize the absorption of sunlight for photosynthesis, and this upright position is possible because that watery fluid in vacuoles increases. You remember what is known as turgidity. When plant vacuoles obtain or absorb a huge amount of water, there is turgidity, and by this turgidity, a weak plant can adopt an upright position, especially to maximize the absorption of sunlight for photosynthesis. Note, the membrane surrounding the large central vacuole is called the tonoplast. This is a special term, the tonoplast, because it is used to mention that membrane covering the plant vacuoles. We don't have any other case of organelles where the membrane is endowed with a special term or a special name. The diagram here shows how big the plant vacuole is. We always remember this detail. In a plant cells, mostly in most cases, there is one big central and permanent vacuole. You can now look for the opposite now to describe the situation. In the animal vacuoles, there will be many small, temporary, and widespread in the cytoplasm. Plant vacuoles contain not only water, sugars, and salts, but also water-soluble pigments and toxic molecules. Remember that a pigment is any substance which is able to provide a specific color. Then, pigments present in vacuoles are responsible for many of the red, blue, or purple colors of flowers and some leaves. That we say flowers and some leaves present the colors we know because of pigments that are present in vacuoles of their cells. The toxic substances help protect a land plant from herbivorous animals. Some plants are not eaten by land animals because of toxins they contain in their leaves, and these toxic substances will be stored too in their vacuoles. The central vacuole functions in storage of both nutrients and waste products. So when old leaves fall off, they go away with the waste products. This is a very important system of excretion in plants, as they don't possess excretory organs as animals do. As organelles age and become non-functional, they fuse with the vacuole, where digestive enzymes break them down. This is very important too in terms of nutrition, because when organelles are broken down by digestive enzymes of vacuoles, they avail food which is going to be temporarily stored in a vacuole. Now we should mention that these two functions are going to be assumed by lysosomes in animal cells. In plants, the central vacuole is also involved in cell growth by occupying most of the volume of the cell. This is because plant cells grow by expanding the vacuole rather than by increasing cytoplasmic volume. Animal vacuoles. Few animal cells contain vacuoles, but fat cells contain a very large lipid-engorged vacuole that takes up nearly 2/3 of the volume of the cell. Here we are talking, for example, about cells of the adipose tissue, that tissue found underneath our skin, used for the storage of fats. The vacuoles of some protists are quite specialized, including contractile vacuoles, for ridding the cell of excess water. digestive vacuoles for breaking down nutrients. Here we talk about, for example, some protozoa. We have examples like amoeba, like paramecium, etc., which are able to pump the excess water outside their organism in order to prevent bursting. Other vacuoles are used for storage or to segregate toxic materials from the rest of the cytoplasm. So when these vacuoles store toxic materials, it is to avoid or to prevent them from dissolving in the cytoplasm, which finally can result into the death of a whole cell. Chloroplasts. Plant cells and cells of other eukaryotic organisms, for example algal cells, that carry out photosynthesis typically contain from 1 to several hundred chloroplasts. Okay, here you should retain that chloroplasts are able to increase their own number by a system of binary fission. Chloroplasts bestow, give, or provide an obvious advantage on the organisms that possess them. They can manufacture their own food. You remember that chloroplasts are present in photosynthetic organisms, make them to be autotrophic organisms. Autotrophic organisms meaning that they are able to make food for themselves. Chloroplasts contain the photosynthetic pigment chlorophyll that gives most plants their green color. Okay, here we should know that the task of chlorophyll is not uniquely to provide the green color. The green color itself is useless for a plant. Chlorophyll is a pigment, a green pigment, which is going to not only provide the green color, but also to absorb the solar energy. The fixation of solar energy cannot be possible without the presence of a chlorophyll. Chloroplasts are bounded by a double membrane, which includes an outer membrane and an inner membrane. Remember that we have only 3 cases of structures in cells that are enveloped by a double membrane: the nucleus, chloroplast, and mitochondria. The double membrane encloses the semi-fluid stroma, which contains enzymes and thylakoids, which are disc-like sacs. In senior 5, you will study that it is in the stroma where there is a long series of chemical reactions that we commonly know as the dark phase, while the other phase, which is the light one, is going to take place in thylakoids, and you have details the next year. Note, a stack of thylakoids is a granum. So remember that a thylakoid is one disc-like sac. So when we have many disc-like sacs together, they make up a granum. Then, as the granum is a singular, the plural will be grana. So when we have different groupings of thylakoids all together, they are grana. But one grouping of thylakoids will be a granum. The diagram of a chloroplast here shows the principal parts of it. You can see the outer and the inner membranes enclosing a fluid known as a stroma. But what is more obvious here, it is the presence of grana. The grana as groupings of disc-like sacs, and each grouping here is a granum. Mitochondria. Mitochondria are found in nearly all eukaryotic cells, including those of plants, animals, fungi, and most protists. The number of mitochondria correlates with the cell's level of metabolic activity. For instance, motile cells, say mobile cells, cells which are able to move, like a spermatozoon, or contractile cells, let us say cells which are able to contract and relax. That's the case of myocytes or cells of muscles. They have proportionally more mitochondria per volume than less active cells. So if a cell is very active, it is supposed to be containing a great number of mitochondria in comparison to a less active cell that is supposed to be containing a less number of mitochondria. This goes or comes with exactly the level of energy required in each cell. The mitochondrion is enclosed by two membranes. The outer membrane is smooth, but the inner membrane is convoluted with enfoldings called cristae. Remember, we say one mitochondrion and many mitochondria. One crista and many cristae. Remember also that a mitochondrion is enclosed by two membranes. That's to mean a double membrane, exactly the same case as chloroplasts. The inner membrane divides the mitochondrion into two internal compartments. The first is the intermembrane space, which is the narrow region between the inner and the outer membranes. The second is the compartment known as the mitochondrial matrix, which is enclosed by the inner membrane, and this is going to be illustrated by the following diagram. This diagram of a mitochondrion shows the presence of two membranes, the outer membrane and the inner membrane, and you can see that between both membranes there is a space, and this space is filled with a liquid, and that space is known as the intermembrane space. Intermembrane space because it is found between two membranes. Also, you can see that the inner membrane is enfolded. It presents prolongations that enter into the matrix of the mitochondrion, and these prolongations, which finger-like projections, they are known as a cristae. And these cristae are going to play a very important role because they are going to provide a larger surface area where reactions aiming the production of energy are going to occur. You can see also that in the matrix, which is an internal fluid, there is the presence of ribosomes, and remember that both chloroplasts and mitochondria are able to produce their own proteins, proteins that will be required in the process of aerobic respiration regarding mitochondria, or proteins required in the process of photosynthesis regarding ribosomes in chloroplasts. Cytoskeleton, finally. The cytoplasm of all eukaryotic cells is crisscrossed by a network of protein fibers that supports the shape of the cell and anchors organelles to fixed locations. This network is called the cytoskeleton. So this will be a network or a web of fibers, of filaments, of tubules everywhere in the cytoplasm, and this will contribute to give a specific shape to a cell. Okay, we know that plant cells have a specific shape because of their cell wall, but we should know also that animal cells possess also a certain specific shape, and this is not possible without what we commonly know as the cytoskeleton. In addition, we should know that centrioles Centrioles are the elements of the cytoskeleton, and they play a very important role in the cell division, especially when they drag chromosomes during anaphase, and details will be discussed in senior 5.